Next up, we have the University System of Maryland. Maryland, it's a Mission 11 flight experiment.
Hi everyone, hope you're all doing really well. I'm Stacy Manuel and I'm a bioengineering major at the University of Maryland. Unfortunately, all my other teammates are off busy being really excellent students and having their internships, so I'm the only one presenting today. But our project is called the Inhibition of Pseudomonas aeruginosa biofilm with Antimicrobial Silicone and Microgravity. And I know that's a really long name, but kind of sums it up really nicely. And we're from the University System of Maryland, which is right here in the DC, Maryland area. And our teacher facilitator is Daniel Serrano. So I'm just going to give a brief overview of our short presentation. First, we have the research background, what we did with our experiment design, basically our current progress, and how this program has impacted us as student researchers. And that is the mission patch for our team, and it's really awesomely designed. It's also designed by one of the UMD students on campus. So first, our research background. So our question was a really simple one. How does antimicrobial silicone affect biofilm growth in space? So antimicrobial silicone has been shown to prevent biofilm growth on Earth with regular gravitational conditions by up to 95%. However, it's not really well studied in space, so we wanted to look at how different materials could affect their growth. So in case you don't know what biofilms are, they're basically just bacteria that grow in clusters. So if you think of plaque, if you don't brush your teeth, like bacteria start to grow. That's basically what they are. And this is really problematic because growing in large groups cause them to be very resistant to antibiotics. So imagine if you have like an implant inside your body. That's not very good if you have biofilms growing on them because that's really hard to get rid of with antibiotics. So the one that we're looking at is Pseudomonas aeruginosa, and that's a type of bacteria that causes a lot of diseases, mostly, I'm sure you all know cystic fibrosis, and that's a very dangerous disease. And it's also been very, very well studied on Earth. So we wanted to look at how it grows in space. So this is a picture of how it grows in space. Basically, you can see that it grows better and faster and in a larger density than it does on Earth. That's because of the low stresses that it has in microgravity. And this was a problem in the Mir space station, which is a Russian space station. And it grew basically in the water containment system. And that's really bad if you have one drinking and like water system. And if you have stuff growing in it, that's not good. So that's also a way that biofilm can play into space travel. So in terms of our experimental setup, we had a FME tube separated into 3 different compartments. So in the first one, we had a freeze-dried sample of our bacteria. In the second one, there's a growth medium where the bacteria can grow, and there's the silicone slides. So I don't know if you can tell, but it's basically 2 different slides, and the first one is a regular silicone, and the second one is an antimicrobial silicone. So we can compare the different surfaces. And the last section has a 10% glutaraldehyde, which is just fixative, and basically what it does, it glues everything together, and so it doesn't change shape while it's going down from space. So on the first day, basically the astronauts unclamp it and allow the bacteria to interact with the medium, let them grow for 2 days, and then the second day they just stop everything, all the processes that's going on, and hopefully that'll maintain the structure and the shape of the bacteria. And we had the same control experiment done on Earth to mimic everything that we were doing in space. So in terms of analysis, I won't go too much into it, but we're looking into the biofilm. So we were looking into getting a matrix stain and looking at the cells. Basically wanted a clear visualization of the biofilm matrices and see the density, the shape, and the thickness of our samples, basically. And in terms of current progress, for our funding, we got $2,125 from our UMD launch project, which is basically a Kickstarter for our campus. And we used that money to go to the launch in Florida last year. And we received our samples from space late last fall. We tried confocal microscopy, which is a type of microscopy that does layers. However, we found that our silicone was actually fluorescing. So we couldn't get that good of a view. So we are currently trying to do atomic force microscopy and electron microscopy to get different angles of approaches. So lastly, just the impact of SSAP. Like Jeff has said, this conference is an awesome opportunity, and I also presented at the International Space Station Research and Development Conference last year. We also saw the launch and saw the cool rocket coming back from the relaunch, the reusability. It was really cool. And we also had a lot of publicity for our experiment, which was awesome. But most importantly, it was the designing experience that was really, really helpful. We've never, as undergraduates, you'd think that we'd have a lot of research experience, but this was very, very different in that we had our own question. We started it out, and we had to design everything from scratch, and implementing something that we learned in our class to something we can actually work on. And proposal writing is very important for future graduate research, and as someone who's interested in going to graduate school, this was very, very helpful. So yeah, thank you so much for everyone who is listed here, and especially the professor who helped me out and providing us her lab space. Basically everyone who helped fund us also. Thanks.
Hi.
Hi.
My name is Augustus, and I'm from Stamford, Connecticut. And I have a two-part question. Firstly, you said that the Russians on Mir had a problem dealing with the bacterial outgrowth, and I was wondering if you knew how they resolved that. And the second part of the question is, how is the ISS currently using antimicrobial silicone to prevent bacterial outgrowths in the future?
Okay. So for the Mir spacecraft, it actually failed, and they found out when it got back to Earth that there was problems inside the water system. Right now, the International Space Station uses basically multi-layer filtering system to get rid of bacteria before it even gets to the water system. And I'm not sure if they're using antimicrobial silicone for bacterial prevention, but we were primarily concerned with what if we were going to use medical implants in the future and stuff like that.
Okay, thank you.
Yeah.
So my question is, what inspired you to choose this experiment?
Okay, so I actually have a funny story about that because I was a freshman in my class, like the Intro to Bioengineering class, and I learned about biofilms, and I thought, it seems kind of interesting, but I wasn't too into it, and my professor was super passionate about it. And then a year later, I found out about the SSCP program, and this was an excellent question to ask. And I was like, oh, this is an awesome opportunity. And now I'm 2 years into it, and I learned so much about biofilms. So that was kind of what got me started.
Thank you.
Yeah.
Stacy, it's good to see you again. I think the last time I saw you, we were listening to Elon Musk at the conference
Yes, yeah.
together. So are you a junior?
I'm going to be a senior next
You're
year.
going to be a senior. So this is what I was saying before. Here is an undergraduate rising senior using exactly the same equipment that you're all used to for research level investigation. And you're now you're setting your sights on graduate school. And I'm sure that SSEP research is going to play a role in that. Yes, there's more college after college. And so I think what's really wonderful about this program, it's a pipeline program. We've got communities with upper elementary school students, middle school students, high school students, undergraduates. And Stacy is a role model for what you can do in terms of research as you move through this pipeline. So thank you for inspiring the next generation.
Thank you.